So welcome to our October Physics Matter Colloquium. So this is the 36th edition of this live but virtual scientific expedition, transcending the frontier of knowledge and bringing light to scientific cases related to research instruments and to the Sesame light source in the Middle East. So this live and open colloquia series is prepared by the Forum of International Physics, the FIP, which is a voluntary association of the APS members who are interested in advancing the knowledge of physics and its diffusion by fostering cooperation and communication around physicists of all countries. So we are sharing knowledge across a wide spectrum of topics and multidisciplinary skills, so that can relate to developing community. So please visit our webpage and you will learn much more about those different destinations. So thanks to renowned international scientists that are teaching us why physics matter. So but this month, we are returning to the past. So in March 2021, actually, it was the very first edition that I was sharing, and we had been shining, so a spotlight on a paleontology with Professor Claudio Tunis, who is from Abu Dhabi, from Abdus Salam. It's interesting because our guest is in Abu Dhabi, so I'm mixing the name, but for Abdus Salam, ICTP, so the International Center for Theoretical Physics in Trieste. So today, Claudio is a panelist with us. So we will introduce the members from our panel. So today, my name is Christine Darv. I work at the European Spallation Source in Sweden. This is the ESS, and I'm the chair of the Forum of International Physics. And with us, we have Jo Lewis, who is working also at ESS for public engagement, and she will be monitoring the question and answer session today. Because today is actually, we have a parallel life and two parallel life events happening. So this is why, she will help me with all those aspects. And in another actually matter of fact, so later you can see as well, thanks to the magic of the internet, the discovery as well of more of the work of Professor Phil Manning, thanks to a MOOC, so a massive open online course that we'll be announcing. So just in 1 hour as well, somewhere else, which is called Accelerate Your Teaching, and so can describe a bit more later on. And somehow the little appetizer that you had today was exactly thanks to Professor Manning showing the use of a light source. So now it's a very exciting and unique colloquia that we will start. So with Professor Manning, who is the chair of natural history and the director of the Interdisciplinary Center for Ancient Life at the University of Manchester. So his research focus is the evolution of life on Earth, which he and his team have integrated with the application of new technique and new technology that are borrowed from physics, biology, chemistry, engineering, and computational science, which is also an important factor for the future. So a part of his research focused on the use of synchrotron light to map and quantify the chemistry of samples from both existing and extinct organisms. So all of that from microbe to dinosaur. So his research have led to significant development as well that could have been and that could be used and have impact in other disciplines, like for instance, from nuclear waste storage to developing advanced material for the aviation industry. Phil has undertaken an extensive field work in North and South America, in the Caribbean, in Europe, in Asia, Africa, Arabic Peninsula, where he is right now, in Australia and New Zealand. And he plays an active role in public outreach program, so disseminating all those information and contributing to open days, but much more. So he has been also writing and presenting documentary for television transmitted on National Geographic Channel, on Discovery Channel, on the BBC, and many other international broadcasters. So he's also the author of book from the children all the way to popular science, promoting the public engagement of science. And he's always working to invest in future generations. So with this introduction, not perfect, but this is really so nice to have you with us on this Physics Matters. So the floor is yours, and pass it on, as you said.
Thank you very much. It's a great pleasure to introduce me in such a very kind way. So thank you. I hope I can at least live up to a little bit of what you've just said about me. But well, thank you. And this evening, ladies and gentlemen, I'd love to take you on a journey. And I'll take you on a journey which is hopefully a good story as well. I know scientists might shy away when I say I'm going to tell you a story, but actually some of the best science, I would argue, is structured and should be a damn good narrative that keeps you entertained. And I think the science that I've been very fortunate to be part of these last few decades has been an amazing journey. And I've learned so much from my students, from my colleagues, and I've learned that science can't happen as an individual. You have to work as part of a team. And so the science I'm going to share with you this evening is part of working with a very large team of, I think, remarkable people. Now, I'm going to see if my the joys of technology work, and they do, which is always wonderful to see. And let me start by shedding light, hopefully, on deep time using imaging techniques which are born out through physics. And I love this image because it just, for me, speaks volumes about what my science had to deal with 100 years ago. We were limited by visible light as to what we could see and understand about the natural world. And for me, just using visible light, it makes us appreciate many of these objects just as beautiful things. But there's so much more we can learn from objects, but through the application of physics. And the team and I who work together are usually associated with digging up giant bones of dinosaurs. So you're already thinking, well, hang on, this talk's going to take us to something about particle accelerators, physics, and synchrotrons. Yes, it is. But the actual raw material, dare I say it, the fossil fuel that drives our science does come from the ground. And how we excavate that material from the ground has changed vastly over the years. The top left image you see is actually a near-infrared laser scan with 3D photogrammetry overlaying, so that you can see dinosaur footprints which are marching off on a Jurassic floodplain some 150 million years ago. But the techniques by which we excavate bones out of the ground really hasn't changed in 150 years. But the techniques which we deploy to study dinosaurs and other life on Earth has changed hugely. When you look at these creatures in a museum, that's usually the first place you'll come across ancient life in some form. It doesn't matter if it's early human remains right the way through to dinosaurs and the earliest forms of life on the planet. It's a museum where you come face to face with these wonderful objects. And don't ever think of museums as these dusty places where specimens go to stand and collect dust. No, this is at the absolute cutting edge of science. These objects have such life which is purely a function of the techniques available to us now, which allowing us to reinvestigate what these fossilized remains mean and what we can learn from them. And there's so much we can learn from these bones. They're not just collecting dust. One of the things with paleontology, which I've always, when I was brought up looking at fossils as a child, I understood the concept of a body fossil. This is the physical remains, the bones, the teeth of a once living organism. And that's something easy to grasp. Trace fossils can be slightly harder because this is literally the preserved remains of an action, a footprint, a burrow. Some even include eggs in this, but there's lots of debate if eggs are body fossils or trace fossils. I'll leave you to decide on that one. But there's a new fossil on the block. These are chemical fossils. This is unequivocally a function of the work that's being undertaken by multiple scientists around the world now, where they're using imaging technology to tease out chemical information that is locked in these fossilized remains of once living organisms. I'd like you to think to that paradigm that we're probably taught, well, I was taught 40 years ago in school, that the fossil is just a mere echo a translation of that original organism into something which we look at today as just being an echo, a ghost of that original animal. It's been replaced, it's been mineralized. Well, that paradigm has shifted again. We're now seeing fossils as potential sources for additional information on the chemistry of that original organism, which can give clues about actual processes which went on when that animal was alive, based on the chemistry preserved in the fossil. Now, it's worth remembering that fossils are actually a very rare thing. And then taphonomy is one of my favorite fields in the field of paleontology. And taphos, nomos, burial laws. This is what happens when you bury something in the ground. And I'll briefly mention, in my very kind introduction, there was a mention of nuclear waste storage, or maybe biohazard storage in the future, where we create unpleasant materials we've got to put in the ground. We really have to understand what happens when something enters an Earth system. And I think have that in the back of your mind during my talk as I take you through this journey of understanding what happens and how things are fossilized. So there we have our life assemblage on the left. We move across to the organisms have died. We have a death assemblage. And then we've got our fossils. And of course, then we collect everything. I wish the fossil record was like these boxes, complete. It isn't. At every stage, there is a process which will take away components within that sort of collective of material. The life assemblage is, as it would suggest, a complete system. But after death, through predation or through scavenging, or even the process of decomposition, burial, exposure, weathering, and erosion, these all cut back into the process of losing information. So what actually gets collected on the far right-hand side there is something which is only a fraction of that original life assemblage, which can be very frustrating for paleontologists. So when we do get information, we have to make the most of it that we can. And whilst we have looked at images of fossils for decades, centuries, as being beautiful objects in visible light, we're now realizing that there's so much more in the electromagnetic spectrum that allows us to investigate some of the most, I think, beautifully preserved iconic fossils that exist in museums around the world. And here we can see Archaeopteryx on the left, one of the first feather found back and described by von Meyer in the 19th century as Archaeopteryx, but specimens from China on the bottom right here from Confucius ornis, beautiful green river formation leaves, and echoes of earliest complex life. And this bottom left-hand corner is one of these rangiomorph structures from Mistaken Point in Newfoundland in Canada. So really, all of these exceptionally preserved fossils have beautiful structure that we can observe in visible light, but is there anything else that we can study? Well, most people, when you start talking about what we can extract from a fossil, immediately jump to the question of DNA. This is usually courtesy of our friends in Jurassic Park, who in the movies were able to extract DNA and reconstruct past life. Unfortunately, DNA is that sugar cube molecule that doesn't really survive more than, well, at the moment, about 700,000 years. That's the oldest DNA sequence that's been discovered so far. It's maybe pushing up towards 1 million, but work by Palbo and other researchers in this particular area have shown that the longevity of this molecule is restricted by its fragility, but its fragility is a function of its structure and how it works within the cell. So DNA is something that might be beyond us for many, many years to come. But maybe that door is not completely closed. But DNA encodes for proteins. Proteins is what the cells are, sort of the DNA is looking to produce. And it's the instruction manual for how to produce an organism, basically. And whether it be, well, sorry, they're probably vegetarians watching. Here we go. There's something for like myself. I prefer to see this. But these proteins are structurally much stronger than DNA and have a much greater chance of surviving into deep time. And there's several studies now showing the longevity of molecules such as collagen. Collagen, which is a beautifully elegant triple helical structure, which is very robust, and has a good chance of survival. And there's been multiple publications suggesting that it's been found in T-Rex from 68 million years ago through to mastodon fossils 0.5 million years ago. And there's lots of discussions about the collagen in terms of whether it had survived or whether there's contamination, etc. But pushing these boundaries to see if we can discover mass peptide fingerprints in this case, suggesting we have proteins preserved, is quite exciting. It means, I hate quoting Jeff Goldblum, but I will, something has survived. And if something has survived in the fossil record for us, in terms of just chemical information, can we extract it? Well, we must remember that we are a bag of chemistry ourselves, as all life on Earth is. And most of it is, I don't mean to be unpleasant, but not very interesting. I'm actually most interested in the trace elements, which are so critical in biological function. Ones that control enzymatic pathways or the actual cofactors which are key in the formation of particular proteins and so on. Because what's really interesting is they emplace themselves in very discrete biological structures, but in tiny concentrations, which are usually beyond the detection limits of most technology. What is really exciting, though, with the advent of synchrotron radiation and the techniques which have been developed herein, there are ways in which you can detect to a remarkable sensitivities, these absolute dilute concentrations of chemistry that could relate to such enzymatic sort of origins in terms of the chemistry. When you look to the fossil record for animals that you might think, well, where do we start? This is how my journey began, because it began over a curry. Now, I'm not eating this particular dinosaur. This is a hadrosaur dinosaur from the American Museum of Natural History. It was dug up over 100 years ago. It's a beautiful mummified dinosaur. I say mummified. The skin has been preserved as impressions, but there could be something more in other mummies. And a few years ago, I had the pleasure of working in the Hell Creek Formation in a locality just near Mammoth in North Dakota. Hell Creek, of course, is the last gasp of the Cretaceous before you get the mass extinction event that killed the dinosaurs. And we're working at a locality just over the border from Montana with a remarkable scientist by the name of Tyler Lyson. And he discovered on his family's ranch a dinosaur like no other I'd ever seen. And I think he did a remarkable job of excavating this quite beautiful fossil. And this is the tail projecting towards us on the left. You can see a limb here. There's an arm encased in this part of the block. And this was the main body block. When this was prepared, you can see Tyler here wearing his hat. And it was a huge task lifting this thing out of the ground. I think this block weighed roughly 10 or 11 tons. And this is really tough work to do. But he recovered it so carefully with an amazing team. And the preparation work that took place on this revealed beautiful skin, that the likes of which I'd never seen on any dinosaur before. And you can see this is the top of the tail and these beautiful ridge-like tiles. It almost looks like the ridge tiles from a roof, but these are keratinous structures that sit on the back of our dinosaur. But I think this dinosaur skin was so beautifully preserved, but when we looked at it, it had depth and structure and form, which didn't look like a skin impression. When you look to the arm, and this is the beautiful skin folds on the arm moving down towards the hand, and you can see a close-up of the hand in the bottom left here with that beautiful little pad on almost the paw of the animal. And when you look at one of the fractures in the arm here, you can see the radius and ulna sat with this skin envelope around the organism. And the scales were three-dimensional in structure. It was absolutely beautiful. So we asked a question, which I think was a fair question to ask at the time. What has been preserved? When you section through the skin, and you can see the depth of the skin here, this is the brown layer delineated by the blue dashed line. When you looked at it under a scanning electron microscope, you could see structure to it still. Were we looking at skin structure? What was there? Has something been preserved that's more than just permineralized skin? And that's where we started looking at different techniques where we could tease out additional information on this remarkable fossil. Now, I always think that techniques, oh, go back one slide, sorry. The techniques available to paleontology today are only restricted by the paleontologists who are either wanting to or not wanting to use them. There is an amazing imaging toolbox, which is present for science to deploy now on the fossil remains of anything from humans back to the earliest life forms on the planet. And these are just some of the techniques that the team at the University of Manchester have been deploying over the last 20 years or so. And the one I'm going to start with initially was one which is not my favorite, because this is a destructive technique, but it's one which is still very powerful. And I don't actually put it in here, I just realized, and it's pyrolysis, gas chromatography, mass spectrometry. But this is what we're looking at here. So there was a sample of scale was taken from this stunning fossil, which I would say if you find yourself in North Dakota, it's in the North Dakota Heritage Center on display. And I think it's one of the most beautiful things on the planet. But when you take a thermal hammer and nudge a piece of the skin from this organism, what was fascinating, when you looked at the macromolecular composition of the scale chemistry when compared to the surrounding matrix, here's the sediment which was surrounding our dinosaur, but this is the actual skin. And we can see a carbon repeat pattern here, which is reminiscent of what we call an aliphatic signal, a signal of indicating we've got breakdown products of proteins present. Now, I'm not saying there's intact proteins here, but there is a discrete group of carbon compounds here, which is indicating we've probably got breakdown products of original organic material that seems to be associated with this particular fossil. And that got us thinking, what else could potentially be preserved here that something can survive for such great depths of time? Not only why has it survived, how has it survived, but how can we use this maybe? We then decided that if we're looking at skin, an incredibly powerful technique that we could ally to the pyrolysis, and a technique I should say is totally non-destructive. We don't have to pyrolyze, burn the sample to push it into the gas chromatography column. Here, there is an infrared beam, this is my colleague, I should say, geochemist, Professor Roy Wigelius, who has just retired, but has also just been made an emeritus professor at the University of Manchester. And he has been one of the pivotal people to drive this research with regards to geochemistry and understanding the preservation of fossils. Here you can see us looking at a fossil leaf from the Green River Formation. An infrared beam is being pushed via a little crystal, a diamond, which goes up against the sample itself. Within the crystal, you get total internal reflectance of the infrared beam. Some is absorbed within the fossil, and what isn't absorbed, you pick back and read, and you see which infrared reactive compounds are basically absorbing different wavelengths of infrared light. And you can correlate those to organic compounds present within your sample. The wonderful thing about this particular piece of tech, it allows you to map across your fossil. Let's look at two rather interesting samples that have gone through this piece of kit. This is one of our PhD students' animals. Sorry, I'm sure it's not one of our PhD, but this is part of one of the shed gecko skins of one of our PhD students' pets. And he very kindly brought the shed skin into the university, and we scanned it. And you can see just to the right of this, there are some really bright colors. These bright colors correlate with the peaks that we're seeing on the right here. And these are amide peaks, which are typical of the amide groups that we find within keratinous tissue, that sort of wonderful keratin, that hydrophobic material that's so important for skin, nail, and hair, and so on. And it is present within, no surprise, within this living organism, albeit it's in the hinge line, we attenuate, we lose some of the amide groups because we don't have as tough a large amount of keratin there. What is remarkable, this image down here is of another fossil that we were working on at the time that's approximately 50 million years old. 50 million years. And what is incredible, it's lost some, but there is still a signal mapping within discrete scale structures, which correlates to the amide groups that we're seeing in the skin of an extant reptile. And this is reptile skin, a lizard skin, from 50 million years ago. So it's showing there is in the just so conditions for preservation, where the taphonomy, the burial laws are in favor of preserving something, that we might get a whiff of some of the original organic compounds that comprise this material. This was really exciting. It was exciting for many reasons, not just that we were seeing survival of an organic signature, one that we could attribute to specific organic groups, but more importantly, it was a non-destructive technique. We did not have to scrape bits off the fossil to scan and understand more about its chemistry. We then thought, where can we go next? What is the next technique that might allow us to give more insight into this fossil realm? And that's where we came to the Stanford synchrotron radiation light source in California. I love this facility. It's a wonderful facility. No one should have a favorite synchrotron because they're all important. And I equally love working at Diamond Light Source and European synchrotron facility in Grenoble. All wonderful, wonderful facilities. I know there's lots of other flavors out there as well, which are equally as important. But I've had such great success working with Uwe Bergman and his team at the Stanford Synchrotron. It's been a wonderful place to work. Uwe and his team were beginning to look at a rather curious palimpsest, a reused parchment, which was turned out was a forgery, but the parchment that had been used underneath this forged illuminated sort of image you're seeing here was actually an original script, which was quite important. Now, this is it under visible light, but let's look at it under, let's give it some synchrotron X-rays now. And suddenly you can see writing beneath the illumination on this parchment. The writing is Greek, and it's the words of the method in mechanical theorem by Archimedes. And many of the sentences here were the first time scholars had got to read it. And the reason you're seeing this is quite fascinating. This is Uwe. We love Uwe to bits. He's wonderful and is a great physicist who's helped us sort of build this capacity to image large objects. The reason we can see the writing in Greek is it's rich in iron. Iron because the Gaul ink that was used by scribes in the 9th century was also rich in iron, because this is chemical warfare from the tree trying to sort of defend itself as a wasp decides to make a home for itself, for its larvae. And the iron which is in the gall is a reaction of the tree. It's a defense system of the tree. But the monks discovered many centuries ago that by grinding these galls down, you can make iron gall ink. Now, that is why we can see the writing, even when it's been scrubbed off the parchment, and it's almost invisible to visible light, but there's just a whiff of parts per million of the chemistry of the gall ink left within the parchment, that when you image it under synchrotron light, you can see it. And this is what our setup looks like at the Stanford synchrotron. I won't take too much time on this, but this is the beam pipe coming in here, and you can see our sample chamber here, and this is one of our detectors. And this is a technique that is remarkable because we can scan huge objects. I mean, the largest object we can take on this particular stage, I think, is 1,000 millimeters by 800 millimeters. That area can be scanned in a matter of a day. Usually we're dealing with microscope slides. We can scan large surface areas on this technique to parts per million sensitivity. But we can tune our way through the different wavelengths or energies of different elements to sort of learn so much about their relative distribution across the surface which we're scanning. So what exactly is the process? Well, we're trying to get a fluorescent yield. We call the techniques synchrotron rapid scan X-ray fluorescence. And so this blue beam is representing our X-ray coming in and nudging an atom in our fossil, and it knocks out an electron from the K shell. And there we see that drop from the L to the K shell, and that symptomatic sort of emission, which is what we're measuring, because that energy from that emission, that fluorescent emission, is diagnostic to the actual element that it came from. So with the correct detectors, we can map and identify and map and quantify the relative position of chemistry across a surface. I'll give you a beautiful example. This is one of my favorites, because my colleague Roy was working up at the synchrotron, and they were offered, Roy Wigelius, the geochemist, and they were offered a musical score from the Stanford Library. It was from Cherubini, the 18th century composer, one of his operas. And he was told that his operas were too long and boring. So he went home, and in a fit of pique, he scrubbed out one of the arias. But fortunately for us, the boot polish he used to scrub it out was slightly different chemistry to the music that was sat underneath. So we're able to literally look through with our X-ray specs. So what you're hearing now, we weren't ever meant to hear. So this was thought lost. And maybe his critics, who said his operas were too long, I don't think they were right. I think it's quite beautiful. So what can we see with fossils, though? We're not going to play music back from our fossils. What we can do is play back their chemistry. And we can look at the relative distribution of that chemistry across that surface. And this is sort of your standard emission lines that we're getting for calcium, iron, copper, zinc. That's the iron K alpha, K beta there. So you get your double peaks, same calcium and so on. But basically, we can map the distribution of chemistry. And this is just one point. But if we can connect those points together, we can get a map. So let's use an example. Our friend here, the beautiful fossil Archaeopteryx from the Solnhofen limestone of Bavaria, approximately 150 million years old. For years, people have argued, even though they shouldn't do, that the feathers had been engraved on and they were fake. No, they weren't. These are real fossils. They're beautiful, delicate fossils of feathers. Why do we know that? Well, let me show you. First, this is what one of our rapid scan systems look like. You can see the beam to the right coming in and delicately nudging every atom across the fossil. And we're giving our fluorescent yield, which we're detecting, and building up a picture pixel by pixel as we move across the surface of the fossil. And so we then window specific chemistry that we're interested in when we're looking at each fossil. So each fossil has its own suite of elements that help diagnose what it is. And here, obviously in this sample, for instance, we windowed calcium, iron, zinc, and lead, and so on. But each fossil is quite different, we should say. And so when we're looking at the optical image, there's our feather impressions. Let's now move to a synchrotron image, one of the way we've joined the pixels together, looking at the chemistry, but in one channel. So it's like you're tuning a radio, but you're tuning not between sound, but you'll be tuning between wavelengths of energy, which is the yield energy, the fluorescent energy for that particular atom. So there's calcium. Now, no surprise, there's lots of calcium in the skeleton. It's you know, an OE hydroxyapatite, calcium phosphate, just like our bones. But it's interesting, the calcium is very similar in concentrations to what we'd expect in modern bone, but then it's in absolute balance with the surrounding environment, which is a calcium-rich limestone. That's interesting. So there's not much what a chemist would say is mass transfer. There hasn't been much or any at all really from this, the calcium into that surrounding environment. So it's almost in balance with the environment. Phosphorus is fascinating. Now, phosphorus is actually quite unusual to find. Plants, this is why plants love fossils. They tend to sort of seek out the phosphorus in the fossils. So sometimes when we're in the field, you see a very healthy looking plant. We always look at each other and think, oh my goodness, is that eating a dinosaur beneath it? In this case, though, the phosphorus is not just preserved, as you can see in the bones. Also, you can see some bits of the bones are not as complete as we thought they were. This one's had a kneecap problem, which has been repaired at some point. We'll get to that in a minute. But do you notice that these fine lines running here, these are the rachoses, the central strut, the barb that supports the feather of each feather of Archaeopteryx. If these were engraved by some forger 150 years ago, whatever, they had to have then emplaced precise 200 parts per million phosphorus within these biological structures as well, which of course is nonsense. What's the most logical solution? The phosphorus hasn't moved. So this is phosphorus from the keratinous material that we see in the keratinous feathers of birds today, but it's been preserved in place for 150 million years. And that's absolutely remarkable. It's remained static for so long. There's interesting things you can find out about the geochemistry of the environment as well. For instance, manganese oxide, pyrolusite, is drawn towards the bones because bones are slightly porous. And they have these tiny little structures, canaliculi, which allow for pore waters to be drawn towards the bone. And we see these beautiful manganese pyrolusite. They look like plants, but they're not. It's these dendritic manganese crystals growing around the bones. And it tells us something, again, about the preservation environment. One of the more slightly disgusting things we might find is curatorial things. I shouldn't say that's disgusting. The next slide is curatorial in that within bromine, we're seeing the bromine used within glues and consolidants, which have repaired this fossil at some point in its life. The bit that is slightly disgusting is those sweaty people who might have been picking up this fossil through time. These are the fingerprints in chlorine. So the chances are that person has held some kind of consolidant and have left their fingerprints on the side of the slab in chlorine. We don't know who it was, but we have their fingerprints. So this has implications for forensic studies, etc., when people are looking at fossils and other objects as well, I might add, which is quite fascinating. But one thing that was a little bit disappointing, and we were hoping we would maybe see a little bit more chemistry associated with these feathers, but they look like feather impressions. So we were pleased to find something, i.e. the phosphorus being preserved. But what I think is incredible is when we started looking at other fossils. Before I do, isn't it interesting to note how many times have you walked down the street and spotted a carcass of a bird that's been kicked around for a while, it's dried, most of the viscera, soft tissue is gone, but it seems those feathers persistently hold on to that skeleton. Those ligaments and tendons that hold the feathers to that bird are really tough. They tend to stay in place. But what is even more remarkable is that all the viscera is gone, but these feathers still look pretty good. Why do the feathers preserve so well? It might explain some of the fossils we see in the fossil record. Now, this is actually a scan going on at Stanford, and this is this particular fossil, Confuciusornis sanctus, this is showing the real-time scanning in high Z. So we're doing the sort of the heavier elements because we can do that through air. When we're looking for lighter elements, the low Z elements, we have to use tender X-rays at a slightly lower energy, well, a lot lower energy, about 3.15 keV. And as a function of that, we have to place this into a helium bag so that we don't get the beam attenuation associated with getting the signal from our fossil that we're scanning. But what was remarkable about this particular fossil when it was scanned is, before I'll tune through radio synchrotron on the right here in a second through the different wavelengths, as it were. But this is a composite map which was produced by Roy Wigilis. I think it's quite beautiful. We've got zinc is in green. It's a rock associated with volcanic eruption, lots of zinc there. You've got calcium, lots of it in the bones, no surprise. But the surprise element that appeared was copper, which is very rich in the beautifully preserved feathers. Much better preservation than we see in Archaeopteryx. But what is this copper? And if we look at phosphorus, again, phosphorus rich in the skeleton, but we go through each element now by each energy. Chlorine, not that interesting. Sulfur, lots of sulfur in the feathers. Calcium, again, in feathers and in the bones. Iron, there's an iron stain around the fossil. Copper is huge peak of copper in the feathers, and so on. So we can go through literally tuning through the different energies to see what chemistry has been preserved within this fossil. So to understand what this chemistry actually means, at the moment, we've just shown synchrotron rapid scan X-ray fluorescence maps. And these are maps showing the relative position, concentrations, and the chemistry that has been preserved. But what does that chemistry look like in detail? Is it original to the animal? Is it something that's been in place during the process of fossilization? What does the chemistry mean? To understand that, we have to deploy a technique which synchrotron users have used for decades at synchrotrons, which is X-ray absorption spectroscopy. Because just in the same way you can tune in to each energy for each particular wavelength of element you're interested in, likewise with the X-ray microscope, you can look at different oxidation states. For instance, in this case, we're looking at different forms of iron. And each form of iron has its own highly distinctive fingerprint. And sometimes we can diagnose if that fingerprint is one of an organically bound element or one which has been introduced as a function of the environment. And that's really important if you're trying to understand the chemistry of a fossil. Is that purely environmental, or is it something even secondary, maybe associated with bacteria, for instance? Or is it actually endogenous? Is it something that belongs to that organism? Now, going back to that copper that we're looking at in the feathers of the Confuciusornis, we're able to look at the spectroscopy of copper. And we found that natural copper melanin from the squid, Sepia officinalis, was able to give us this beautiful, it's one of the purest forms of sort of this melanin. And we were able to compare that with the melanin, the copper that was preserved within Confuciusornis, other feathers as well that we had from the Green River Formation. Just look how different it is in terms of especially the outer shells here with regards to a copper oxide. It's very distinctive, the eumelanin configuration. We're also able to diagnose that it was light element coordinated. We had a central absorber atom, there's our copper, and we have oxygen-nitrogen coordination, so light element coordination in that first shell, and we've got our eumelanin monomers attaching around there. And we realized that what we're looking at was, with that copper, was in fact melanin pigment. And this chemistry was we think, a good marker for melanin. And we didn't just stop by saying, okay, we found it at the synchrotron. We remembered that Fourier transform infrared spectroscopy can also help us map organic functional groups. And that's precisely what we did. And we looked at the melanin molecule, you can see on the right here, and we're able to map and associate specific chemistry from the fossilized feathers that we were looking at and correlate that to specific structures that we'd expect within melanin preserved within these fossils. Again, this was work done by Holly Barden, Roy Bergelius, and a much bigger team of folks that is feeding into this research. What did this mean when we're looking at the actual reconstruction, say, for Confuciusornis sanctus? Well, it meant that we could understand something of the distribution of melanin pigments across the organism in relation to high concentrations to low concentrations. It's giving you sort of a dark and light map across the organism. It's not going to give us anything beyond that. We can't tell you the color. Color is complex. Color is tantalizingly complex. It's just beyond our reach on this using these techniques, for the simple reason it's a function of how light is absorbed, reflected, refracted within a material. So structural elements. It's also what you eat, depending on what time of year, what food you've got on your diet. There could be different pigments that can be distributed through keratinous structures like feathers, which would alter the color. And likewise, there's iridescence and other structures which are also quite hard to map. But these are being looked at by scientists now, by paleontologists. And I'm very excited. I think the future is, I don't want to use the pun bright, but I think it'd be much more brightly colored. We also looked at briefly the Archaeopteryx feather, this first feather described by von Meyer back in the 19th century. And it's a beautiful feather. And there are other studies. There was a wonderful study by Carney that was published in Nature, looking at the melanosomes, which are the organelles in which pigment is manufactured. You could do it as point analysis across the feather. The wonderful thing about the technique that we developed at Stanford, it allows us to map the concentrations across 100% of the fossil. Whereas if you're looking at a scanning electron microscope to map every single melanosome, the user would go bonkers after a while for the simple reason you'd spend a year in the ESOM just mapping the whole feather. This technique takes literally an hour to map the whole feather at the synchrotron. And what it allowed us to see as well is that, again, we had that very distinctive peak of melanin, which was present at the tips, and especially on the leading edge, we found, of the wing. Why is that important? Well, there's many flighted birds today that have concentrations of melanin pigment on the leading edge of the wing as an adaptation to strengthen the wing, because melanin, in the same way it strengthens your hair when you go gray like me, and the hair becomes brittle, when it's melanized, it's much stronger. So it's a very useful adaptation for flight on the leading edge of a flight wing. And I will take you to the last bit of research on my last slide that we've been working on with regards to pigmentation at synchrotrons. And this is one of my favorite little mice. This is Apodemus, which is a beautifully preserved mouse, approximately 1.5 million years old. And you can see just a faint hint of its ears here. And it's wonderful. You can see the phosphorus ears in blue down here on the bottom right-hand side. It has very pretty ears. But what is fascinating about this, because we can use X-ray absorption near-edge structure spectroscopy, XANES, and we can look at the specific oxidation states of sulfur. We could map the organically bound sulfur across the skin, ears of this beautiful fossil. What is really fascinating is when we looked at the sulfur and its coordination with other trace metals present, especially zinc, it looked like it was bound to the zinc within the first shell, which is fascinating because that is something that we see in fair melanin pigment. So it's got quite a heavy element coordination, having sulfur in that first shell. And we would like to suggest that what we were seeing in this particular fossil was the presence of fair melanin pigment, this blonde, brown, reddy brown pigment that was preserved in this wonderful fossil. And again, this was due to the remarkable spectroscopy being undertaken by the team at Manchester Stanford Diamond Light Source, and of course my relentless colleague, who is so important for this research, which is Roy Wigelius, our geochemist at Manchester. And last but not least, and this is one of my, again, I've got lots of favorite images of fossils we've scanned, but this one I particularly like because I think the caterpillar who produced this structure, these are frass tubes. It's basically caterpillar poo. And the caterpillar poo is showing what it's digesting. It's chewing the leaf. And as it's chewing out the leaf, it's producing poo. And it produces poo in these, we call these things frass tubes. These frass tubes actually puts the pellets around it, so it helps protect the caterpillar as well. And this one clearly was quite a smart caterpillar because it spells out the word CSI, which I think is quite wonderful. But what it's doing is eating part of photosystem 2, because the plasticinin, which is a key cofactor in photosynthesis, which is rich in copper, is present within the leaf. That's what you're seeing here, mapping within the leaf. And because it doesn't want to digest this is pushed into its poop, and it goes into the frass tubes. But you can see what it's been eating across this leaf and hyper-concentrating it into its frass tubes there. So we're learning something. What does that tell you? Well, it tells us the leaf was green when this animal was feeding. So if you're looking at the season, the time this leaf fell from the tree, that could be interesting for some paleobiologists who are interested in seasonal implications for a particular locality and so on. And I think it's just a wonderful example of how this technique can be deployed across many parts of the animal and plant kingdom and beyond. I'm not sure if you can actually read that because of the thing on the top of my screen. I'm not sure if you're seeing it, but in all honesty, when we went to the synchrotron, very few of us could actually really have predicted what we were going to find. And I think the words here of Isaac Asimov, that great moments in science are rarely met with the word eureka, but usually by the phrase, that's funny, is one of those moments that we had at the synchrotron. So many times we would get signals back from fossils that we had to then go back to look at extant analogues or related organisms to try and make sense of the chemistry we were seeing in the fossils. And it's binding both the modern world and the ancient world, that's helping us better understand the preservation of material. But more importantly, it's helping us understanding the mobility of material within a burial environment. If you want to put something in the ground today, you want to make sure it's going to stay put if it's particularly unpleasant. But if you want to run an experiment for 100, 1,000, or 1,000,000 years, we have a horrible habit of dying. So how do we do this? Well, we can model it, but how robust are those models? Well, maybe we should be looking to the past, because the hindsight the fossil record gives us is a series of time-stamped material that's been put in the ground at specific moments in Earth history, and we can then map and understand that process of mass transfer from organism into the environment and from environment into the organism. And where we do see cases of non-mobility, understanding why those biomolecules have remained immobile and whether we can use that understanding to better understand burial environments for the present and the future. And I would also argue it can help us react to disasters, natural disasters, and ones which we have a hand in as well, such as the Deepwater Horizon spill, when hydrocarbon-derived porphyrins are pumped into the environment and get into the food chain and are eaten by animals such as the brown pelican here. And they try to get rid of these toxins, pushing them into their feathers. And rather than having to kill the animal to work out what it's suffering from, a single feather, as it pushes the chemistry, as the calamus grows in the skin, you can detect the levels of hydrocarbon-derived porphyrins and understand how it's impacting different species across quite a range. So I think there's huge implications for using synchrotron-based imaging as well for understanding modern populations in the natural world. And this work, as I've said many times, can't happen without an amazingly large team of wonderful people. And these are just a few of the key people who have really helped us over the years. And on that note, I'd just like to thank you for inviting me to speak this evening. And I'm hoping if there's any paleontologists watching, if you've not used the synchrotron already, please use the synchrotron. It opens your eyes to a world which I didn't even believe was possible. Thank you very much.
Thank you so much, Phil. A wonderful talk, so many different topics and interesting ideas and thoughts, and this lovely picture you've painted of how you can start with a series of questions or perhaps a that's funny, and by collaborating with different people, find new technologies to start answering those questions. I think it's fascinating. So now is the time for some questions and talks. We have also Claudio Tunis, who's joining this panel. So feel free to ask questions along paleoanthropology, as well as whatever the very long description Christine gave to the work you do, Phil. So from dinosaurs also to kind of studying modern, old but human objects as well. There was a question I saw on the Q&A from one of our people who's watching. Yongzhang, would you like to answer the question out loud? I can give you the microphone. If you'd like to do that, you can type in the Q&A or the chat if you'd like to do that. Let's see if I can find you, and then I can give you the microphone if you like. You've got the option to talk now if you'd like, Yongzhang. I'll tell you the question out loud while we work out if this is going to happen. So the question was seemingly simple question, but maybe with a complicated answer. If you're doing this work with synchrotrons on fossils, ancient fossils, does it damage them? Can the X-rays can the beam damage the sample?
That is a very good question. And in the synchrotron rapid scan X-ray fluorescence, where we're moving the object very rapidly in relative to the beam, the dwell time on a single pixel, a single point on the fossil is enough to excite the chemistry, so we can map the chemistry there, but it doesn't damage the specimen. However, if you've got material which is rich in copper and you're doing something, one of the spectroscopy techniques where you have to have the beam on a single spot for 20-30 seconds, you might see photo reduction of the copper. And so you'll see the spectra change live as you have the beam on the sample. This is usually the case with extant samples. The geological samples and paleontological ones tend to be much more robust. But so there's the spectroscopy, we have to be acutely aware of that. And are you able to visibly see something when the fossil comes out of the beamline? No. The exception to that case, I think, is amber. And I think there's been some studies with amber which have been scanned at the synchrotron, and it has changed the color of the amber as a function of the X-rays going through that material. But I believe that that is reversible. And but at the same time, I would still consider this technique a non-destructive technique.
Okay, thank you.
Thank you.
So, Claudio, as well, I wonder if maybe the two of you could have a small discussion about the difference, if you like, between wanting to look at things which are 100 million years old and looking at something that's perhaps a few hundred years old. What would be, are there ways that one can give information about the other? So if you are looking at something that's of a human time, can it tell us about fossils, ancient dinosaur, perhaps fossils, or the other way around? Can these dinosaur or million-year-old fossils tell us something about human time?
Oh, we lost.
Oh, we've
There's your answer. I would say we've been looking at injuries in dinosaurs, and the injuries in dinosaurs are fascinating insofar that they show distinct chemistry associated with healing. And there's a suite of cofactors in healing in humans that we seem to, bone is, it seems remarkably conservative. And so dinosaurs were healing from injuries using a suite of chemistry, which involves copper, then zinc, and then the final remodeling by osteoclasts using strontium. And we can map the presence of this chemistry in pathologies in dinosaurs. And we've also noted this in mammal bones as well. So we'd imagine there's some really fascinating work to be done on pathologies in humans from the fossil record, or from the anthropological record, as it were.
Did you want to add something as well, Claudio? You're back with us? We can't hear you right now. Let me see if I can unmute you. If you could.
Okay,
Yes.
Can you hear me now? Yes.
Yes, thank you.
Just a small comment. First of all, thanks, Phil, for the beautiful presentation, and was really very exciting discussion. And I think you have shown in general that what you said at the beginning about the role of natural history in connecting, for example, the human cultures that we have on the planet, so different and so divided by religions and ideology. I think natural history is now, with this new scientific enrichment, with the new methodologies for studying natural history, really will have a big role. And I wonder, also, for example, in museums, how can we embed in the specimen that are shown in the museum, in paleontology, paleoanthropology, all the science now that is giving us these beautiful stories, as you said, about the ancient humans, but also about dinosaurs and so on. So these are two general comments. Concerning the damage, I am aware that for example, X-ray microtomography with synchrotron radiation can really produce damage in on the DNA. So then it becomes difficult to make the sequencing of the DNA. So one must be very careful, and actually, you need to develop some protocols in order to make all the different analyses. And I think you should collect the specimens for DNA analysis before doing the X-ray microtomography. And I remember there were a couple of papers on this by the groups, for example, from the European Synchrotron Radiation Facility. And also, for example, well, I'm familiar with the X-ray microtomography, but another example is you can destroy the signal that is used in electron spin resonance dating. So if you take, for example, a Neanderthal tooth that you would like to use for producing a date based on electron spin resonance, with an X-ray microtomography, you can really kill the signal. So these are two examples coming from my kind of discipline. I would like to ask you also something else. Now, paleontology, but also paleoanthropology are really producing big data because, the analysis, DNA analysis, and the chemical analysis, peptide analysis, protein analysis are producing this big data. And I'm not familiar with the use of artificial intelligence and deep neural networks and so on. What is happening? I think there is some beginning in the use of artificial intelligence to analyze data, but I'm not familiar if that is happening also in paleontology in general.
It is. First, I'm extremely jealous of the fact you keep saying DNA. I would love DNA. DNA would make my life so much easier. Having a molecular phylogenies would make the record a dream, but we don't have that. So it's really frustrating. So you're absolutely right to say that. And yes, of course, it does denature that material if you put it under intense X-rays. So that's thank you. That's very important. In terms of your second question, the jet lag is hitting me
again. I think that the oldest specimen are about I think in paleoanthropology maybe 500,000 years, I think, with DNA in the Neanderthals from Spain, where they got the DNA, but not for your paleontological specimens.
No, it's true. And please remind me, your second question was?
No, was the one related to the use of artificial intelligence?
Oh, artificial, yes. We're using machine. Yes, we are using machine learning in tomography. Interesting, because as you work in tomography, it's a good one to use. We're fascinated by the structure of bone, and my PhD students, who are wonderful, hate segmenting samples.
Yeah, that's right.
Anyone hates segmentation. It's the part of tomography which, if there isn't an algorithm which can do it efficiently, it's a problem. So we're looking at machine-based learning approaches to improve segmentation and to search for solutions to allow segmentation. We're also using machine-based learning and genetic algorithms in computational multi-body dynamics, where with my colleague Bill Sellers, where we're creating three-dimensional skeletons in a physics engine, and where you have a skeleton which you rig your muscles to, and then just as a piano, when it plays music via holes in that piece of paper that goes through the piano with those automatic pianos, the muscle activation system, when it's activating the muscles, is like music. If it's not choreographed properly, the muscle groups don't function, the animal doesn't move forward. But once that choreography is perfect, we use evolutionary algorithms to search for optimized muscle activation patterns. And this way we can better understand locomotor mechanics of extinct animals. So the the power, the computational power available to us now is changing the science and how the science is actually being done in paleontology. And I think it's a really exciting area. And I think that particularly in locomotor studies, we're learning so much more about the biomechanics of extinct organisms as a function of the computational capacity being made available to us. And also with some of the modeling that we're doing as well with regards to bone behavior is really important. So we can create some very large models now, but we need even more and more processes to run those models to better understand the mechanical behavior of bone. So we can maybe extract clues on the behavior of biomaterials, which can be used to make modern materials for industry. And I think the field of biomimicry has been with us a long time. It hasn't kept up with imaging. And I think imaging technology now is going to, there will be a revolution in biomimetics, because we have got the ability now to look at resolutions in organic structures that wasn't possible 10 years ago when biomimetics was the thing. Now I think there's going to be a revolution in paleobiomimetics, because we've got a depth of life to plumb, to search for structures that are engineering solutions to complex problems which life has overcome 100 million years ago, or maybe even 500 million years ago. But the technology is now there for us to get that information. That's exciting.
Thank you.
We've had another question on the Q&A, perhaps for both of you, which is close to my heart as well. The question is about which tools we're using. So you've talked about X-rays, but what about things that X-rays find harder to see? The suggestion was protons or perhaps proteins, but things with hydrogen or other things in them that maybe X-rays aren't the right tool. How do you handle looking at things in fossils where X-rays might not be the best tool? Are there other
It's interesting. Yeah, we've been using for carbon compounds, which are obviously quite important for organic compounds. I've worked with an amazing group from Soleil in Paris, and it's using Raman spectroscopy, basically, X-ray Raman spectroscopy at the synchrotron facility there. And I think the actual environments in which we're scanning as well can be improved to get the lighter elements. In terms of neutron imaging as well, I think is an area which I think is ripe for exploration by paleontology and other disciplines. But I would just hope that these techniques, well, I'm not currently playing with proton or neutron imaging, but I've got colleagues who have been playing with these techniques. I think it's rich territory there, would be my answer to that.
So do I. There's another question, I think, here. Anna, that was just thank you for your answer there. I had a short question for you, Phil. Have you got like a shelf with all your favorite fossils lined up that you want to look at in the future? And if you have, then what's the next one on the shelf that you want to study next? What's your next exciting after the mouse with the cool ears? What would your next one be that you want to look at?
It's a difficult question, but my next exciting thing I'm really interested in is getting kids interested in science. And that for me is as important as any fossil I get to cuddle and hold and close and hug. I love these objects. They're beautiful. And I want kids to fall in love with the natural world again, just as I did as a child. And I think the role that museums, universities and schools can play in getting people to love the natural world again, so we can have more people who grow up to become scientists, poets, scholars, people who just look at the natural world and go, isn't it beautiful? Because if we don't love the natural world, we won't look after it. So we need people to fall back in love with planet Earth. And if you want, but that's what I've been working hard with the Oxford Cambridge Examination Board with their new Nat History and GCSE in the UK. That's a big thing, and they're amazing what they're doing. and the fact that kids in the UK in 2025 and be able to study natural history O level, or GCSE, I should say, I find that wonderful. And that's as wonderful as any beautiful fossil I'd like to study.
Quite right. Maybe I'll just take this moment to have a couple of minutes about the course that Christine mentioned at the start, that you've been kindly helped support and contributed to. So anyone watching who is a teacher of kind of what I call secondary level, so 11 to 19-year-old students, there's a new online course which Christine and also Phil have contributed to. It's with a number of partners, including some science partners with CERN, ESS, and also the Copenhagen University, but also pedagogy, so teaching partners. So a number of universities, Malmö, and the teaching part of Copenhagen University too. And also some science centers in Lund and Denmark, Sweden and Denmark. So this MOOC is for teachers who are interested in accelerators or particle accelerators or machines like synchrotrons or neutron sources, which have accelerators perhaps behind them. And it shows different ways that you can use those stories, those modern, exciting descriptions of how science happens in the 21st century. It's not one man in a small room with a blackboard. That's not how science happens these days. So if you want to tell those stories and use those stories to teach the things you do teach, like the structure of an atom, or how are molecules arranged and how do we know, or perhaps a bit about the physics of how these machines work, then this online course could be for you. And you can have a look at some of the have I even got it on this this QR code behind me? It's very small, but it might be one of those, but I think Christine has shared it in lots of the normal places too. And Phil is on one of the videos as part of that. It's called Accelerate Your Teaching. You can always just Google that. It's through the Scientix team who run all sorts of courses for high school teachers. I also, if we have, we've lost Claudio, and I think we've now answered all the questions. So I will just take a moment to remind people what the next talk is going to be in this program of events. Let me share the right one. So next month's talk, if you can see the right one there, is by a colleague from the UK called Professor Carsten Welsh. He's actually German, but he has worked in the UK for a number of years. He's going to be talking about accelerating science and society, how accelerators are used in society. Carsten Welsh has also contributed to that online course that Christine and I are involved in. So we're really pleased that he has also agreed to give one of these next talks. That's going to be on the 30th of November at the usual time, Thursday at 1600 Central European time. There's a QR code you can scan behind me, or perhaps next to me, or on the screen. And if you have signed up to this, you might even get a reminder email about it. So don't forget that next talk, but let's just take another moment to thank Phil once again for a really fascinating talk. Thank you so much, Professor Phil Manning.
Thank you very much. It's been a pleasure speaking to folks. Thank you.